much for inviting me here. Also, thanks, Harry, for introducing the subject of general anesthesia. So I have to confess, I'm a physiologist, so a lot of the things that you will see are physiology recordings from the brain of mostly animals, actually, that lose and recover consciousness from anesthesia. And I know most of you are not physiologists, but I think it's informative to try to understand what kind of things can we measure about the brain and what could they be telling us about loss and restoration of consciousness. And I think we have a much easier time than our neurology colleagues because our patients don't have damaged brains in some sense. They come into us with normal brains. So perhaps if we could understand how consciousness is lost and recovered in a normal brain, maybe we can say something about how it doesn't happen in a damaged brain. So I want to show you a movie that I think addressed a question that was raised earlier. And if any of you have to undergo anesthesia soon, I'm sorry if this will freak you out. But what you will see is a person who is under general anesthesia, okay? They're intubated, they're on a ventilator, and they're paralyzed, except for their left arm. And what you will see is quite dramatic. And this is a movie that was donated to me by Ian Russell, and I can't thank him enough for this.
Eve, open the fingers of your left hand.
This person is under anesthesia. Surgery is in progress.
If you're feeling comfortable, Eve, open your fingers again. And close them again. Now this time, if you are in pain, if you have pain, and only if you have pain, open your fingers. That's very good, Eve. Okay, can you just open your fingers again now? Just open your fingers now. And close them again. That's good.
Are you sufficiently freaked out?
If you can hear my voice, open and close the fingers of your left hand. That's fine. If you're in any pain, could you squeeze your hand twice? If you're comfortable, could you squeeze your hand once? That's fine.
Okay, so just note that surgery is taking place, right? And this person, except for their left arm, is completely paralyzed. Now, how often does this happen? And if you listen to Ian Russell, you would be very scared because it happens much more than 1% of the time. In some studies, depending on anesthetic, as many as 30% of the time. And this happens with modern anesthesia techniques as well, although this has been started in the 70s. So I know Harry talked to you about measures of depth of anesthesia, and I'll say a lot more about that. The question is, can you put an EEG monitor, process it somehow, and predict when these people are conscious? I think if this was a person after brain injury, you would definitely rule out the possibility of vegetative state, right? I'm not a neurologist, but it seems pretty obvious that they have some level of consciousness, right? And the sensitivity and specificity of BIS, again, this is the measure that Harry talked about, is lousy. You know, Ian Russell has been doing these experiments since the 70s, and people took real objection to this because they said, well, most of these people don't remember anything. As far as they're concerned, nothing happened. They don't have any recollection. And people were really interested in saying, well, how often does anesthesia with recall happen? Meaning, I ask you after the surgery, do you remember anything? And how often does that happen? Well, that happens about once in 1,000 cases, let's say. It depends a little bit on how the study is done and how it is defined. And this is prompted recall, meaning you're specifically asked. But the conclusions still stand. That is, if you try to guide your anesthetic delivery by this measure of depth of anesthesia, or you don't, and you just use sort of standard clinical doses of anesthetics, there is no difference. So about once in 1,000, there is anesthesia with recall. And indeed, some patients have a terrible experience, and they have pain, and they have this fear of being paralyzed. People describe the sense of being dead and all sorts of awful things. But surprisingly, not all of them, and this is consistent with what people described in the locked-in state. Some people don't have a terrible experience, and they're not traumatized, but clearly some fraction of people who experience awareness, especially with recall, go on to develop post-traumatic stress disorder. So that's not good. So again, I am a physiologist, and I will work in animal systems, so I will try to understand what actually happens in the brain as the brain recovers from anesthesia, and maybe that will help us come up with a better way of detecting depth of anesthesia and somehow calibrating to the brain activity such that consciousness does not happen. Right? So what is depth of anesthesia? I mean, it's in some sense similar to assessing patient's level of consciousness, perhaps in a neurological setting, but because we're inducing this state through pharmacology, there is sort of a dogma in our field that depth of anesthesia is directly related to the amount of drug in the brain. The more drug you have in the brain, the more deeply anesthetized you are, and in the deepest planes of anesthesia, you will see physiological patterns that are quite similar to coma. They will progress through the ones that are kind of similar to slow wave sleep, and eventually towards the awake patterns. And an ideal depth of anesthesia monitor, it's been argued, should track this concentration of the brain. So what does this mean? Really, if you unpack this a little bit, you see anesthesia as sort of pharmacology-only state. That is, the level of consciousness is in some sense controlled by the number of receptors that are occupied by the drug. The more receptors are occupied, the more unconscious. The fewer receptors, the more conscious. And as you would expect that the drug concentration should change smoothly and gradually, one can naively expect, and so should the brain. So in the first part of the talk, I will sort of challenge this notion of pharmacology only. Pharmacology is important, but what I will show you are some very non-trivial dynamics in the brain under anesthesia. And in the second part of the talk, I will try to argue for a measure of this brain state. How can we measure it? And there are many theories out there and many ideas about how to do it, and we have our own pet theory, and ours is much less ambitious. And all of this will be done in animals, so I'll try not to wade into consciousness too much. So, but this is my very simple-minded view of recovery of consciousness. I mean, consciousness is clearly a miracle, but recovery of consciousness is sort of a separate miracle, right? You can think about the number of different states that the brain can produce, and no matter how you define them, you very quickly arrive that that number is absolutely astronomical. It is so large that no single brain can even sample a small fraction of all possible activity patterns in 1,000 lifetimes. And while we don't know why certain activity patterns are consistent with consciousness, it's clear that that's kind of a special state of the brain. If I at random chose an activity pattern, it would be very unlikely that this would give rise to some sort of reasonable experience. So that's why it's not surprising that we can perturb consciousness in any number of ways, using any number of different drugs that Harry showed work on a number of receptors, circuits, etc., and disrupt consciousness. But when you recover consciousness after anesthesia, you don't just become conscious, you return to being yourself, meaning your memories are intact, your personality stays more or less the same, cultural influences and whatever else, you recover to being yourself. And that's a very unlikely state of the brain. So you can formulate this recovery of consciousness as some sort of a search through this enormous space to find those patterns that are meaningful. So, and again, I'm a physiologist. So for me, deep unconsciousness is something like this, right? This is a suppression that Harry showed. This is characteristic of deep anesthesia. It is also present in severely disorganized brains like under coma, hypothermia, encephalopathy, anoxia, postictally. It's really a broken brain pattern. So, and if you record in the awake brain, and again, Harry showed you this, these happens to be recordings in rats, but you would get the same thing pretty much if you look at the human EEG or EEG of any other vertebrate, really. So to me, recovery of consciousness is sort of how are these blue squiggly lines morphed into the red squiggly lines? If we could understand that, perhaps we could understand something about recovery of consciousness. So, well, we have to represent them somehow, and there are many ways of doing this, and many people have shown you similar kind of slides. You can compute a power spectrum, and without getting technical here, the only point is that there are many fluctuations in the signal, and a power spectrum sort of parses it into different frequency buckets, right? How much oscillations occur at this frequency, at that frequency, so on and so forth. And a couple of things to note here, that oscillations are across several orders of magnitude in frequency, and that one can distinguish the brains from a deeply anesthetized from an awake patient, at least in the case of this drug, although I will talk about some exceptions to this in the second part of the talk, quite readily. So instead of now drawing these squiggly lines, we're going to be looking at power spectra, and we can see how the spectra change over time as the animal emerges from this deep comatose-like state to an awake state. And of course, the spectrum, as a disclaimer, doesn't describe the whole system. But where do we want to put the recordings? I know Nico knows and loves this slide very well. Clearly, there are some better targets than others. Central lateral nucleus of the thalamus is known to be involved in anesthesia, sleep, coma, etc. Cingulate cortex has figured prominently at this conference, and retrosplenial as well, cingulate and retrosplenial. So that's where we're going to record simultaneously, and our experiment is as simple as can be. We start in this pattern of burst suppression, we wait a while, we lower the drug, we wait a while like that until the rat wakes up, and wakes up for us means it gets up and tries to run away. Okay, so how does this happen? So, well, this is really the major, that's the answer essentially. So on the blue trace here shows you the anesthetic concentration. And in these colorful plots, you're seeing how these spectra change over time. Now, the more power is shown in red, the less power is shown in blue, X is time, Y in frequency, and these are all the different channels. So a couple of remarkable things to note here. Certainly, it is structured. This is not some sort of a random noise. Two, it is not gradual. There are abrupt changes in the signal. You can see all of a sudden something changes and we have some onset of power in this particular frequency. Sometimes you can see that these things percolate the entire thalamocortical system, and I'll show you some examples of this. Sometimes the thing I want to point out is that there is also no one-to-one correspondence between brain activity and anesthetic concentration, right? So this is, notice this is time in hours, okay? This is a really long and boring experiment. So we're looking here, concentration is fixed, but the brain flips and flops between different states. So we want to understand this, but first I just want to show you an example of what this actually looks like, right? These are recordings simultaneously from the thalamus, central lateral thalamus. This happens to be frontal cortex. None of this is critical. Animal is sitting at a fixed anesthetic concentration. It's doing its thing here, delta-like activity, and then all of a sudden, abruptly, clearly the pattern switches, right? And now you want to ask, well, what happens first? A lot of people have focused their attention on this, and with neurophysiology, we can answer that question unambiguously. It's at exactly the same time, or it can. Right? This is sort of a global state change in the thalamocortical system, not entirely surprising given how interconnected thalamus and cortex are. Okay, so let's return back to this point. So this is really sort of complicated, but it's structured, so we can maybe take advantage of the correlations that are present in the signal and reduce the number of dimensions here. And without getting into technicalities, we can apply standard techniques like principal components analysis, and we can capture about 70% of the variance in just 3 dimensions, right? And that helps a huge way if the brain is trying to navigate this enormous space of activity, because reducing the number of dimensions clearly shrinks the space, right? So that's one part of the answer. Now, what about the second part of the answer? So now that we've reduced the number of dimensions, we can ask, well, where is the system in this space? So we can basically plot a histogram of the data projected onto these dimensions. So this happens to be in the first 2 dimensions, fixed anesthetic concentration, but brain is not fixed, right? It can be found in one of 2 such states. So what happens if I change anesthetic concentration? Okay, so I have different states. Some disappear, some appear, but there is no one-to-one correspondence. Again, again, again. Okay, so not only are the data confined to a low-dimensional space, but within this space, the data are confined to some patterns of activity that are stereotyped, and they're discrete. So can we find them? And yes, we can, and this is a result of clustering analysis. Oops. All right, there you go. Now data projected in the first 3 dimensions, doesn't really matter. We find 8 distinct clusters that are shown by these ellipsoids, and each one of the colors shows the different kinds of patterns. Blue means different ways in which the system can exhibit burst suppression. Green, delta waves, kind of more consistent with lighter planes of anesthesia, let's say more similar to sleep, and red are theta and gamma oscillations, okay? All right.
Something about your dimensions. Are those just arbitrary dimensions you got on?
Formally speaking, these are eigenvectors of the covariance matrix. It's basically, yes, I should have shown the slide. I have, yes, so these are kind of typical spectral shapes, if you will. These are kind of characteristic shapes of spectra. I can show you the data afterwards, but yes, they're meaningful. These are not arbitrary. This just, you know, if you have data on X and Y that are correlated, it doesn't make sense to plot X and Y. You can sort of rotate the coordinate system, so you're plotting it, say, diagonal versus another diagonal. That's what this is. It's a sort of simple mathematics trick. But I was kind of trying to spare people as much physiology as I could, but this one I probably should have kept. Okay, right. So how can this happen, right? How can the system be confined to just these small substates? Well, so here is an example of the distribution of the data, distribution of states shown in red. And in green are distribution of the amplitude of fluctuations, okay? And if you superimpose the two, you can see quite convincingly in many cases that there are rims of green surrounding peaks of red. And what that means is that when the system arrives into these densely occupied states, the fluctuations die down. So what am I saying? In so many words, I'm saying these are attractor states of the thalamocortical system, okay? So now what are these states, right? So we can look at the spectral profiles of each one of these clusters and ask, what do they look like? And that reveals more structure. So we can identify 3 clusters that are characterized by burst suppression. You can see in every one of the channels there is low frequency oscillations below 1 Hertz that characterizes burst suppression state. Second group of clusters, we see delta waves that are everywhere. And in the third group of clusters, we see theta oscillations, okay? But the individual members are distinguished by how the power in higher frequency ranges distributes among different channels. In some cases, we can see the cortex sort of activate, but not the thalamus. In this case, we can see thalamus, but not the cortex. And in this case, we can see the whole system come online, okay? And the reason why there is a red asterisk here is this is when rats wake up. Not to say that every time they come here, they wake up, but every time they wake up, they were here, okay? So now we can simplify the whole thing by looking just at the sequence of states. As the animal wakes up, now they're in cluster 1, now they're in 2, then now they're in 19, whatever. So this is an example of that thing happening for a single rat. As the anesthetic concentration is high, then we have more burst suppression. You lower it, you get deltas. You lower it again, you get theta gamma. That's not surprising. What's surprising is that not all transitions are equally likely. For instance, you never bubble up to the green states for any one of these other blue states. And the same thing can be said about from green to red. So we can calculate all of this by computing probabilities of transition between all pairs of states. And you can see that, well, probability of staying in the state where you are, shown here in the diagonal, is the highest. And that confirms our results with stability. That's not surprising. You can see that the squares shown in white are the transitions that we have never observed in about 36 hours of recording. So maybe they do happen, but they're vanishingly rare. And other transition probabilities, they're not all the same. Some are much higher than others. This is on a logarithmic scale. So some of them are 2 orders of magnitude higher than others. Okay, so we can now put it all together and now represent each cluster by one of these spheres. And the transition probability is the arrow that links them, right? And this really reveals sort of the dynamical structure of how consciousness gets recovered, at least in rats, from anesthesia. And what you can see is that certain places in this network play special roles, like this one and this one. They're hubs, meaning there is no way to avoid coming here first, then coming there if you ever want to reach the awake cluster, okay? So you can stare at this long enough, and we can prove this using some straightforward sort of analysis of connectivity here. So what did I tell you? I tell you that the brain activity is confined to a low-dimensional subspace. In this subspace, the data are clustered into certain distinct activity patterns, and only certain transitions between them can exist. As a result of this, there's sort of an ordered sequence, meaning you have to be in place A in order to ever get to place B. Not to say that it's the same sequence all the time. The transitions are stochastic. So, and for me, this is very similar to what, say, a computer has to do when it has to recover from a brain-dead state. When the power dies, there is no information that survives in the computer, yet the computer is able to restore itself, to sort of bootstrap itself. And I think what this is telling us that the brain has some fundamental capacity to restore itself, and that is separate from the question of what constitutes consciousness per se. This is more of a mechanisms of restoration. So that's the first part of the talk. So why does this matter? Well, you know, should we think about recovery of consciousness as far as depth of anesthesia is concerned on a continuous scale if the brain dynamics are not continuous, they're clearly discrete? And 2, usually we compare features when we say the person or an animal is more deeply anesthetized. Does this pattern of activity resemble more the awake or the anesthetized pattern? But really, if not all transitions are possible, perhaps we should be thinking about it more sort of in graph theoretical sense, if you will. How many hops away are you from being awake? And I think that might change the way that we think about measuring depth of anesthesia. And of course, my hope with all of this is not to understand how anesthetics work. I really want to understand how people recover, say, from brain injury. And just in this context, and this is at the level of sort of, I don't know, a dream perhaps, is that this is work by John Victor and Nico showing recovery, EEG during recovery of consciousness in a patient triggered by zolpidem. To me, the very fact that you can perturb a brain-damaged patient years after injury and trigger them into consciousness might suggest that it's not that the brain is not capable of consciousness. Perhaps it's not capable of recovery. And again, that's a separate problem. And in my dream world, that's because they're stuck in one of these sort of attractor states, because maybe the link to the next one is broken. So what do I have to support this? Well, very little, except for this study again, where Nico and Jonathan looked at fluctuations in coherency of the EEG, a different measure than what we looked at. But nonetheless, the point is that when the subject was emerging from this minimally conscious state into a state of higher consciousness by zolpidem, there were abrupt fluctuations in the EEG, suggesting that there are these state transitions, maybe. So that would be something I'm very much interested in. So just want to switch gears very quickly and say that, well, the quest for neurophysiological correlates of unconsciousness is not new. This has been done from the very beginning of electrophysiology, and this is classic 1930s paper by Gibbs, and Lennox showing the slowing of the EEG with increasing doses of ether. And that's great, only it's not true for all anesthetics. And just like with sleep, REM sleep was called paradoxical sleep. It's not paradoxical in any fundamental sense. It just doesn't fill this dogma. So we have drugs like ketamine, we have drugs like nitrous oxide, et cetera, that don't produce these patterns, right? So, and that's why depth of anesthesia monitors don't work, because in one way or the other, they look at this kind of feature. And that's not surprising, because this looks at activity in each part of the brain in isolation. This doesn't tell you how the brain works together. Okay, so sort of a new sort of thinking about this is look at connectivity, and I know many people have shown connectivity, and many people have in the audience have done connectivity analysis. Melanie, for instance, done some excellent work on this. Just want to show a couple of examples here. This is from Emery Brown's group showing the frontalization of EEG shifting in the alpha oscillations towards the frontal part of the brain under propofol anesthesia. This is also beautiful work by Anchal Lee and George Mashour's group showing feedback connectivity that is universally disrupted under different anesthetics, notably with ketamine. And that was a big breakthrough, because ketamine is a known outlier. And that's beautiful work. My issue is that while it tells you what correlations exist in the data, it doesn't tell you how the system works as a whole. Right? So what we wanted to do is try to start from another direction. Ask what should be the requirements for the brain to be conscious. And our claims are much, much, much more modest than, say, Giulio Tononi's claims. We're not saying that this is consciousness. We're asking what kind of features should the brain possess in order to enable consciousness. So much more modest claims. And here we thought that stability of cortical dynamics is quite relevant. So if the brain was a very stable place from a dynamic standpoint, then any perturbation would damp out, be that perturbation imposed by magnetic stimulation, sensory stimulus, effect of one part of the brain on another part of the brain, it would just quickly die. And that's not really consistent with an awake brain. If it was too unstable, then any perturbation would blow up without bounds and eventually resulting in some sort of an epileptic-like state. And that's obviously not consistent. So this leads us very simply to hypothesize that perhaps stability of the brain is important, and the brain ought to be able to control its stability to some optimum range between the damped and the unstable fluctuations. So that's what we hypothesized. So we, some kind people in Japan donated us ECOG data. These are electrodes that are sort of evenly spaced, covering the entire hemisphere, 128 channels. They were very kind to give us the data. So what does it look like? Well, it looks like a bunch of squiggly lines together, and we want to understand them together. We don't want to look at any feature of any one thing. We want to look at it together, and a standard way of doing it is sort of describe the evolution of these signals using our regressive technique. And none of this is very important, except for we do this separately for small chunks of data. So we allow the data to globally be nonlinear and no stationary, but we approximate it locally but a linear and stationary system, and that has many mathematical advantages. Why? Because the stability of this system is totally understood. Okay, this is very, very simple. And again, without going into the details, this has to do with eigenvalues of this matrix. If this parameter lambda is less than 1, oscillations damp. If it's more than 1, they explode. If it's close to 1, they're critical. So where are they in the brain? Okay, so this is the distribution of eigenvalues in the awake monkey before it got drug, and you can see clearly it is close to 1, and it is close to 1. Now monkey gets some cocktail of drug. We've done it with ketamine alone, ketamine medetomidine, propofol, and we've done this on humans, ECOG. All results are the same. The critical modes go away. Okay, it doesn't matter what kind of drug you get. Ketamine, propofol, all the same. Different monkeys, this is highly statistically significant. So this is at the face value consistent with our notion that stability is an interesting thing to look at. Okay, so but how does stability come about? What is it a feature of? So we wanted to keep everything about the signal is the same except for the global properties of the system. So what do? We just take a number that is actually from a Gaussian centered at 0, so on average we don't do anything at all, and we move the signals with respect to each other by a number taken from this Gaussian distribution. So all the signals are the same. In fact, all pairwise correlations between signals are also the same, minus the arbitrary time shift in the correlations, and only the global correlations go away. So what does this do? Well, I forget already which one is which. The point is that this is a very, very subtle change in the signal, right? You cannot tell the difference. I mean, I can't. But stability analysis does, and this is what we've done. If this thing detects consciousness, then we ought to be able to put that in statistical terms. Can I reject the null hypothesis that the monkey is awake while it is awake, using the real data as opposed to the surrogate data? Okay? So this is the distribution for the real data for each one of the different monkeys concatenated across different experiments. So if you want to reject the null hypothesis that the monkey is awake with any statistical confidence, you would have to wait 1 in a million would do it, right? And well, this one is a bit noisy, but for most of them, the distribution is quite tight, meaning you're very unlikely to reject the null hypothesis that the monkey, to falsely reject the null hypothesis the monkey is awake. Now, the red distribution is for the time-shuffled surrogates. So although all the features are the same, stability analysis, A, they're different from real data, and B, they're much less consistent. And this, again, suggests to us that maybe there is a tuning that the brain does in order to keep itself in this sort of dynamically critical state. Now, just to tie this to connectivity, I mean, there are many ways to express and think about connectivity. So, but autoregressive matrices also give us a notion of connectivity because they sort of summarize how electrodes influence themselves and influence other electrodes, okay? So we can just basically project these autoregressive matrices onto the brain. This is what we see in the awake brains of a monkey. We see that connections mostly originate in primary sensory cortices and go towards the front. This was done totally blindly, just by fitting the numbers. We had no idea where this is coming from, but this in some sense recapitulates what we would expect for sort of flow of information from the occipital sensory cortices towards the front. And this is what happens with different drugs. In propofol, connections go away. I know Melanie and others have spoken about propofol becoming sort of a state of disconnected brain, and yes, I think that is very true, according to us as well. Ketamine is quite different, right? We can see that the connections reverse direction, right? And they go starting from inferior temporal towards the occipital, amongst other places. It is kind of interesting to note that ketamine is a hallucinogen. So it is an interesting thing to think about, that perhaps the activity in the visual cortices is now driven by other parts of the brain rather than being driven by sensory experience. So, but the point is that the stability is preserved, the results with stability are consistent, whereas just about everything else we look at is not, meaning the spectral changes are different, the coherency is different, the frequency dependence of stabilization is different for different drugs, but the stability itself seems to be robust. So I think these results have pretty interesting implication, or at least a connection with the work that Marcello has shown. And in fact, I think they've motivated our thinking about the system in this way, in that what does it mean that a system responds differently to a perturbation depending on the state of the brain? That means that the stability of the brain is different in these different states, right? And I suspect that we could make some pretty quantifiable predictions to the results of TMS experience without actually doing them, simply by analyzing the stability. If in the awake brain there are many dynamically critical states, you would expect that the perturbation would last a while, and it would be complex because there will be many modes that are perturbable. As more and more modes fall off the sort of dynamic criticality range and become stabilized, fewer and fewer modes will be recruited by the perturbation, and the perturbation will be simpler and will die down. So that's certainly a prediction we would make based on these results. It would be very interesting to see if we can actually predict the results of perturbation given our analysis. And so what did I tell you? I told you that we have a kind of again, a very simple theory that has to do with what consciousness is. It tries to establish what are some basic dynamics that could allow it to happen, whatever it may be. And I think we can argue about information integration theory and details of it, but it is probably true that consciousness is sort of an emergent property of the whole brain. It's integrating information in some sense from various sensory streams, from internal representations, whatever it may be. And it seems to us that in order for this integration to happen, dynamics have to be confined to this sort of a critical range rather than stable or exploding. And that's, and another thing, just sort of a side note, that I think what's interesting about our way of thinking about this is that we can connect the connectivity view of the world, what is correlated with what, to the dynamics of the system as a whole. And I think that's an interesting way to think about the system. So with this, I just want to say thank you. Drew Hudson and Paula Calderon and Don Pfaff at Rockefeller. These are the guys who we did all the experiments on rats with. The dynamical criticality, I want to thank Naotaka and Toro and Riken who gave us the data, Marcelo and Guillermo, and well, actually two Guillermos, for helping us analyze the data. Some of this is published. This was the human stuff. The monkey stuff is still, getting rejected from one journal after the other, but it will get somewhere. And this is the rat stuff, and of course the kind people that gave us money. And thank you very much.